Hello, welcome everybody. And thank you for coming here to our first in vivo seminar in this celebrity series. And this is a very unique event because it's really the first time we're all here together in this hall that all of those amazing scientists were giving talk before, and we're having another amazing scientist that is coming for today. And we actually see that it's a real human. It's breathing. It has thermogenesis, so to me it marks also to coming back as a community of people that actually listen to each other and not do laundry while listening in the other ear for a seminar. As I said before, if you want to talk to the person sitting next to you, just don't forget to mute yourself. So today we have a very special guest, which is Alan Saltiel, that is coming from UCSD. He's a pioneer in the field of cellular metabolism and energy metabolism, and I hope that he's going to tell you a little bit about his career, because his career is very unique. Where he started in Duke University as a zoology student, and today he is getting close to 350 papers with about 20 patents. He started his career with a PhD in North Carolina, University of North Carolina, in biochemistry, and then he moved on to industry, where he was the senior director of the Department of Cell Biology at Park Davis Pharmaceutical Research Division, which then became a very unknown company called Pfizer. So and then from there he joined the he joined the University of Michigan, and where he was the director of the Life Science Institute at the University of Michigan. Today he's at UCSD, as I said, and he's professor of medicine in UCSD, and he's also the director of the UCSD UCLA Diabetes Research Center and the UCSD Institute of Diabetes and Metabolic Health. And today he is going to tell us about new insights into the regulation of energy homeostasis. And so please, Alan, thank you for coming.
Okay, thanks, Orion. Hopefully I'll be able to see the screen, right? What? Be sure to share screen. We're okay. Can I get that in? Yeah, something on there. All right. Oh, I have to look at myself though. I'll take off my glasses. I can't see. So can you guys hear me okay? I hope everybody can hear me on Zoom. I guess they'll tell me if they can't. You can lower that too. What? I can get rid of that? Get rid of that. Yeah,
you're breaking. My Wi-Fi is not that great.
Oh, good. Okay. All right. Okay. All right. Anyway, thank you very much. Thanks very much, Orion, for inviting me. I have to say, I've never been introduced before as a real human. I've always wondered if I was a real human, so I'm glad you confirmed. And really, anyway, it's great to be here, and it's great to be able to see all my friends and colleagues, especially in our diabetes center, which is really a terrific, I think one of the great diabetes centers in the country, and I'm really proud to be a part of it. So again, thanks a lot for the invitation. I'm looking for there you go. Okay. So when I started as a fellow in Pedro Quattrocasas's lab, well back into the last millennium, I won't tell you what year it was. I was interested in trying to understand how metabolically active cells like fat, liver, and muscle cells responded to hormones to make a decision whether to use or store energy. And, you know, I thought this would take me a couple of years. I'd wrap this up, then I move on to something else. And, you know, I was disabused of that idea pretty quickly. And I realized that this new field of signal transduction, which was emerging at that time, was a little more complicated than that. And then we also learned, I think, over the years that these cells didn't just respond to hormones, but also talk to each other quite a lot by making their own hormones and metabolites that had a big effect on systemic metabolism. But you know, over the years and over that time, at that time, and continuing to today, I was basically a reductionist at heart, which is a kind of a nice way of saying a dinosaur, actually. And I was really interested in understanding how cells responded to fasting and feeding. Now, you all know the cells respond to fasting hormones like glucagon and epinephrine by activating catabolic processes in the cells, in fat cells like lipolysis, in liver cells, glycogenolysis, and we studied that a little bit. But I was most interested in the feeding part of it. And of course, the granddaddy of the feeding hormones is insulin, and insulin is the major anabolic hormone. So I would say over the last 40 years or so in my lab, we've studied mainly insulin action and tried to understand how insulin works. And this has been divided broadly into three areas. One area is trying to understand how insulin controls the uptake of glucose in cells, especially in fat and muscle cells. And we've studied the signaling pathways that link the insulin receptor. I wonder if I can, let's see if I can get that laser pointer. The pathways that study the insulin receptor to GLUT4, which is a glucose transporter that's involved in these processes in muscle cells and fat cells. We've studied all these signaling pathways. We focused mostly on the G proteins that are actually at the nexus, at the intersection between signaling pathways and the trafficking pathways that are involved in GLUT4 control. And I'm not going to talk about any of that today. Okay, so this is one area. It's still a very active area of investigation in my lab that I'm not going to discuss. A second area of investigation in my lab has been to try to understand what happens to hormone action in obesity. Now, a lot of you know that obesity is associated with reduced responsiveness of cells in the fat and liver to hormones, especially insulin, but also other hormones like catecholamines. And we've been trying to understand how obesity does this. We focused our efforts on trying to study inflammation. We think inflammation is a primary way that obesity represses sensitivity to hormones. And we've been looking at the intersection between inflammatory pathways and the signaling pathways that control metabolism. I'm not talking about that today either. Okay, this is what I usually talk about. This is the biggest area of investigation in my lab. I'm sorry if you're disappointed that I'm not going to talk about that today. You're not going to hear about that. I'm happy to answer any questions. But instead, what I'm going to talk about is a third area of investigation that we've been working on for actually quite a long time, and that's to understand how energy is stored in cells, particularly the storage of energy as glycogen. Now, I know a lot of you are thinking, glycogen, come on. What could possibly be interesting about glycogen? And isn't glycogen already solved? Okay, and I know that's what most of you are thinking. And so today I'm going to spend the entire hour now trying to convince you that, first of all, it's not solved. There's a lot we don't know about glycogen. I'm not going to solve it for you, so don't get your hopes up too high. But there's a lot we don't know about glycogen. And secondly, yes, glycogen is really interesting, at least to me. Maybe this is a bad sign about me, but glycogen is really interesting in that I'm going to try to convince you today that it's more than just an inert form of energy storage in cells. It is actually a cue to the cell and tells the cell when it's a good time or when it's safe to store energy in some contexts or use energy in other contexts. So another way to say this is, and what I'm really trying to hammer home today, is that glycogen is a signaling molecule. Okay, so that's the message today from my talk. Now, so I'm going to go back now many years, and first of all, to give you a little bit of background for those of you who don't know about glycogen, you probably know that glycogen metabolism was worked out in the 40s and 50s by the Coreys, who discovered glycogen synthase and phosphorylase. Synthase is the enzyme that makes glycogen. Phosphorylase is the enzyme that breaks it down. Krebs and Fisher in the 1970s and 80s worked out the regulation of these enzymes. And we know that insulin promotes glycogen synthesis. It's an anabolic hormone. It does this by activating synthase and inhibiting phosphorylase, whereas counter-regulatory hormones like glucagon and epinephrine do the opposite. They stimulate the breakdown of glycogen and they prevent the storage. And we know that protein kinases and phosphatases are the key regulators of this process. Phosphorylation of synthase inhibits it, and insulin promotes the dephosphorylation of this enzyme. Phosphorylation activates it. And this is actually a general theme in metabolism. The metabolic enzymes that are anabolic are activated by dephosphorylation. The ones that are catabolic are activated by phosphorylation. Okay, so this is a general theme. Now, it's a little more complicated than that, especially in liver cells and muscle cells. We know that glycogen is also controlled allosterically by glucose metabolites. And when glucose comes into cells, it's converted by hexokinase or glucokinase to glucose 6-phosphate. And glucose 6-phosphate is an allosteric regulator of these two enzymes. It activates synthase, and this is a forward loop, and I'll be talking a lot about that today. We know that the fasting hormones like glucagon and epinephrine activate pKa, which inhibits synthase and activate phosphorylase through kinase activities on these enzymes. But the action of insulin is a little more complicated. It works in several different ways. Insulin increases glucose uptake to increase the levels of G6P to allosterically activate this enzyme. And insulin also inhibits the activities of protein kinases that inhibit the enzyme. It inhibits pKa, GSK3, AMPK or AMP activated kinase. And I'm not going to talk about the mechanism of this today. This is all well known. But work going back to the 1990s actually has shown us that any of these effects of insulin themselves are insufficient to explain the mechanism of the full activation of this system. And work that we did in the 90s and work from Kai Sakamoto and from John Lawrence and Dario Alessi and others have shown, and Phil Cohen and Patricia Cohen have shown that there must also be a dephosphorylation step that's activated in response to insulin. And Joe Larner told us back in the 70s, and other people have shown, and we've shown, that the protein phosphatase, a dephosphorylate synthase, is the enzyme protein phosphatase 1. Now, this is where the first enigma comes up, because PP1 is a promiscuous enzyme. If you just assay its activity in cells, it'll dephosphorylate almost anything. So there must be a mechanism to explain the very selective effect of insulin on these events in cells. And we thought about this back in the 90s, actually. And two postdocs in my lab, John Printon and Matt Brady, thought, let's try to find a targeting protein that might specifically explain the ability of insulin to regulate the phosphorylation events that occur at the site of glycogen itself. So John, who was a very good scientist, took advantage of a new technique that was just being described at that time called yeast 2 hybrid screening. Some of you were around at that time and you remember this. A lot of you weren't not around. You've never heard about this before, but yeast 2 hybrid screening was a very effective way to identify interacting proteins. And John made a yeast 2 hybrid library from 3T3L1 adipocytes. This was a great library. We used it to screen lots of different protein interactions and found lots of genes. Remember, this is before the sequencing of the genome that we did this. So this is really the cloning of a new gene when we found this. And they used PP1 as bait to try to find a targeting protein. And they identified this gene encoding this protein that we called PTG. which satisfied a lot of the criteria that we were trying to identify to explain the specificity of these events in cells. Now, it turns out that PTG is a member of a family of scaffolding proteins for glycogen and PP1. And all these proteins have a common overall structure. They have a binding site for PP1. They have a binding site for the targets of PP1, glycogen synthase, phosphorylase kinase, a bunch of other proteins I won't talk about today. And they have a module in the protein that binds to glycogen, a carbohydrate interacting domain. And they're all the same. We studied this in the 90s. We made mutations in these proteins. We made chimeras. I'm going to spare all of you all the biochemistry that we did. But it's very clear that these are very important proteins to target phosphatase 1 and the kinases and other enzymes that are involved in the regulation of this process. In fact, the structure shown here, the structure, a few groups, Garcia Jimeno and Hurley and other people have solved the structure of these proteins, of the targeting proteins, identified these discrete, unique interaction sites. Now, the interesting thing is that PTG and these other proteins have very profound effects on glycogen metabolism. This is an experiment. There were experiments done in the late 90s and early 2000s by Matt Brady when he started his own lab at Chicago, who was again the postdoc in my lab that discovered this protein, and Chris Newgard, who did this experiment when he was still in Dallas. When you overexpress PTG in cells, you dramatically increase the glycogen accumulation in these cells. This is an experiment that Chris did when he used adenoviral expression to overexpression in liver in a mouse. It basically turned the liver into like a glycogen sieve. Okay, so they get a lot of glycogen. So these enzymes these proteins have a big effect on glycogen metabolism. So we wondered whether they were regulated, and this could possibly explain some of the regulation of glycogen metabolism by hormones. So we looked at the level of expression and stuff like that of these proteins. And here's an experiment we published a couple of years ago. We turned again to the mouse. We looked at the main paradigm that controls glycogen, fasting and feeding. And we also looked at the impact of high-fat diet on this process. So the first panel here is looking at the glycogen levels in the white bars in normal diet mice, fasting and feeding. You see the well-established big increase in glycogen levels in liver in these mice. And when you put mice in a high-fat diet, you see there's a small increase in the fed state, but in the fasting state, there's a pretty big increase in glycogen levels. This is something that we didn't appreciate at the time, actually, but it's actually increased in obesity. And we looked at the PTG levels. This is the gene, PTG gene. It's called PPP1RC3, 3C. And you can see that PTG gene expression tracks well with glycogen. It goes up in feeding, and it goes up in high-fat diet. This is the mRNA. Here's the protein down here. You can see barely detectable in the fasted state. In normal diet mice, it goes up in feeding, and it goes up a lot in high-fat diet. Now, the interesting thing was that PTG protein and mRNA expression also tracked well with this protein SREBP, which also goes up in fasting and goes up in feeding and goes up in high-fat diet. Here's the mRNA level for SREBP. Here are the protein levels, again, fasted feeding, and you see it goes up in the basal, in the fasted state, in high-fat diet, and in the fed state. And it also tracks well with activation of S6 kinase, which is shown here. You see, which we know actually goes up in high-fat diet mice, even in the fasted state. So we wondered whether SREBP might be connected somehow to glycogen and PTG expression in this paradigm. We were very excited when we looked at the promoter of PTG. Oh, again, this is just for me to remind you that SREBP expression is actually controlled by the S6 kinase pathway. So when you activate mTOR and S6 kinase, this leads to activation of the expression of SREBP, and this is well established. So we looked at the promoter of PTG, and we saw a very good SREBP binding site in this promoter, which is shown here. We did some promoter activity assays in cells. If we overexpressed SREBP, we see an increase in PTG levels. These are just mutants. We could also activate the mTOR pathway by knocking out the TSC gene, which gives rise to this is a gap for a protein called REB, which regulates mTOR. And if we knock out TSC, we also activate SREBP expression. And you see an increase in the expression of PTG. This is blocked by rapamycin. By the way, this explains this long-held observation going back to the 80s from John Lawrence and other people that rapamycin blocks the activation of glycogen synthesis in response to insulin. Nobody ever understood that. This is the mechanism, actually. And we also, that was all done in vitro. We also looked in vivo. So we made TSC, we made a liver-specific knockout of TSC, which gives rise to a liver with constitutively active mTOR. So you can see the knockout of TSC. In these mice, there's, not surprisingly, this is just, if you look at control, the wild type and the knockout, there's an upregulation of SREBP protein and an upregulation of mRNA. And then if we inject it into these mice, shRNA against SREBP, so if we knock down SREBP in this knockout background, we see that this increase in PTG, which is induced by activation of the mTOR pathway, is completely blocked in SREBP knockdown cells. And this is just the same thing as looking at the mRNA. And in fact, if we look at the glycogen levels in these cells, in these livers, you can see that knocking out TSC, activating the mTOR pathway, activating the synthesis of SREBP gives a big effect on glycogen, which you can block if you eliminate the SREBP in the system. So again, what we think is this pathway is getting activated in high-fat diet, and it gives rise to the activation of PTG, which controls the levels of glycogen. So hepatic levels of glycogen are increased in obesity, and the expression of PTG is driving the increase in glycogen, and that's down. Oh, is that me? It didn't offend anybody. That's downstream of the mTOR pathway. So anyway, it looks like SREBP through mTOR controls the expression of PTG, which controls the regulation of glycogen metabolism in liver. So the question is this important? So we made a knockout mouse of PTG. This is a whole body knockout. This took us a long time. I know I told you we discovered these things in the 90s. It's 2021. What took us so long? It took us a long time because it was, for a lot of reasons I won't tell you about today. It was hard. But nevertheless, we achieved a knockout of PTG. We looked at the glycogen levels in the livers of these mice. This is a whole body knockout. There's normal diet and high fat diet and wild type mice you see in white to knockout mice, big reduction in glycogen levels. Okay, not surprising. We knew that PTG is important in glycogen metabolism. The surprise came when we looked at hepatic fat in these mice. As you can see, this is just a measure of hepatic fat. It goes up in high-fat diet. As you all know, high-fat diet induces steatosis in mice, and it's reduced by about half in the PTG knockout mice. This is some H&E staining. You can see steatosis in wild-type mice. Response to high-fat diet, it's reduced a lot in the knockout mice. And likewise, SREBP levels are also reduced in response to high-fat diet. And the targets of SREBP, like the fatty acid synthase gene. Now, I know this is confusing because I just told you that SREBP and mTOR and SREBP controls PTG, but now we get rid of the PTG gene and we're actually having a reduction in the activation of SREBP. What's going on here? We looked at the protein levels. Again, you can see the PTG levels. This is a knockout of PTG. And when you knock out PTG, this is a normal diet and high-fat diet. You see that the elevation of SREBP and high-fat diet is reduced, and activation of S6 kinase is reduced. So the mTOR pathway is reduced as well. This is just a quantitation of these results. So this is a summary of what we're seeing. What we have here is a forward loop. Okay, we think that obesity gives rise to activation of mTOR. This is through hyperinsulinemia and lots of amino acids, inflammation, glucose, other things that are happening in obesity. This initially leads to increased expression of SREBP. SREBP now drives lipogenic genes, but also drives the expression of PTG, which gives rise to increased glycogen. And then glycogen is somehow initiating a forward loop that gives rise to further activation or sustained activation of mTOR, so that SREBP now can be elevated at sustained levels to give rise to steatosis, that is sustained lipogenesis. So another way to think about this is that glycogen is a cue to tell the cell that we have enough glycogen. We now like to store energy as lipid. Okay, it's like there's a threshold level of glycogen. Once you hit the threshold, it's telling the cell shift to lipid. Okay, so it's cueing the cell to shift to lipid. Now the question is, and we don't know the specific answer to this yet, so I'm going to give you a few hints. The question is, what's the sensor for glycogen that's telling cells to shift to lipid? And we don't completely have the answer to this yet, but we think the sensor is AMPK. Okay, and why do we think this? Well, we know AMPK is a sensor of energy status. We know that when energy levels in cells are high, AMPK activity is low. When energy levels go low, AMP levels go up, and AMPK activity goes high. But we also know from the work of Graham Hardy and others that AMPK has a very good glycogen binding site that represses its activity. This glycogen binds in the beta subunit. The structure activity relationship of glycogen itself to the binding site is well known. The structure of the binding site is very clear. We know that glycogen, and it dominantly inhibits AMPK activity. Furthermore, we did RNA-seq on our livers from the knockout mice. And we know that in the PTG knockout mice, there was dramatic downregulation of lipogenic genes with an increase in lipid oxidation genes, as well as the activity of the proteins encoded by those genes. All of this is associated with higher activity of AMPK. And when we measured AMPK activity, it was higher in the knockout mice. So we think AMPK, it's not, maybe not the only mechanism, but we think AMPK is the sensor. Furthermore, we know from studies that we did earlier that high-fat diet and all paradigms of high-fat diet in mice and even in patients leads to a decrease in AMPK activity. This is shown here. This is from a paper we published a couple of years ago in Science, when we were looking at the link between AMPK and apoptosis in liver, and I'm not going to talk about today. We also know that if you inhibit AMPK in cells with insulin, which is something we know quite well, we do it all the time, you can block that if you knock out PTG. So you need glycogen to be able to see the inhibition of AMPK in response to insulin. And then finally, there's a very nice study from Ruben Shaw, where he put mice on a high-fat diet and then overexpressed a constitutively active form of AMPK, which is resistant to glycogen inhibition through the beta subunit. And in these mice, AMPK can burn fat, as you can see here. This is the control, and this is the overexpression. So you can overcome the effect of glycogen by giving constitutively active AMPK, which will repress the mTOR pathway. So again, here's what we think is happening. We think there's a forward loop. When mTOR gets activated by obesity, or in feeding actually, gives rise to S6 kinase, induces SRABP. This induces the expression of PTG. This gives glycogen, and then glycogen is sensed by AMPK. Glycogen inhibits the activity of AMPK. Okay, but that now permits the further active, and now you lose the ability of AMPK to block mTOR, so you get the further activation of the mTOR pathway. This is another way to look at this. The same thing, we're looking at the cycle here where we get obesity giving rise to this pathway, SRABP generates PTG, you get a lot of glycogen. Glycogen now inhibits AMPK and now blocks the ability of AMPK to repress mTOR, to increase the oxidation of fat by repressing ACC, and to actually repress inflammation. I haven't had time to talk about that part of the story today. So glycogen really is a key regulator. It's a key cue in liver to really control the expression of the lipogenic effect and allow for the accumulation of steatosis in the context of obesity. So it's a signaling molecule. So let me now switch to another topic, which I think is even more interesting, and this is about what's happening in fat. So my lab has been working on fat cells forever. We're kind of in a adipocentric group. We think fat cells are at the forefront of sensing and responding to energy needs. And we think those cells are very interesting. And all of you know that fat cells respond to fasting and feeding by dramatically expanding and contracting to control energy needs and provide energy to the tissues. And we also know that they respond to different kinds of metabolic stress. They respond to anabolic stress like obesity by expanding and then kind of staying in a locked anabolic state. They no longer respond to hormones very well. That's because of inflammation, as I told you before, but they kind of stay locked on in the anabolic state. And in contrast, they also respond to prolonged catabolic stress. Now, you all know there are different kinds of fat cells. There are white fat cells. There are brown fat cells that are there mainly to generate heat in response to cold. And then there are these third kind of cell called the beige cell. These are cells that arise in depots of subcutaneous fat that can convert to brown-like or beige cells that express UCP1, the uncoupling protein, that uncouples ATP synthesis to generate heat. And I think all of you probably know the story very well about the beiging of fat cells. So we wondered whether glycogen might play a role in any of these processes. Okay, now why do we think this? Well, it turns out that fat cells have all the goodies to make glycogen. Okay, you can't find a lot of glycogen in these cells, but they have all the enzymes that make glycogen. And in studies that we've known about this for a long time. And in fact, John Lawrence, the late John Lawrence, who's my good friend, and I used to talk about this all the time when we weren't discussing Duke basketball, which we talked about a lot, but when we were talking about metabolism, we noted that fat cells are unbelievably responsive to insulin with respect to glycogen synthesis. You could get hundreds of fold increase in glycogen metabolism in response to insulin, way more than you see in muscle or liver. And so that was kind of weird because where's the glycogen in these fat cells? And Matt Brady actually had published this experiment where he kind of did a survey of glycogen levels in adipose tissue. He noted that there's a little bit of glycogen in epididymal fat. This is in visceral fat. It might be regulated a little bit in fasting and feeding, but if you compare it to liver, look at the scales, you see that there's about 1,000-fold more glycogen in liver compared to white fat. So basically the glycogen in white fat is negligible, right? Even though you can make it, it's just kind of not doing anything. It's barely there. And liver, you see it's regulated, it goes up in fasting. But what about brown fat? Well, Matt looked at brown fat and he saw that it wasn't really regulated a lot in fasting and feeding over time. But look at the scales, there's actually a lot of glycogen in brown fat. In fact, it's comparable to what you see in liver. This is something I don't think a lot of people appreciated. So this made us wonder, well, maybe glycogen is doing something in beige fat, right? We know it's not doing much in white fat. We looked at white fat. We looked in the context of obesity. We haven't really seen any changes in glycogen. But then we decided to turn our attention to the beiging of fat. So there are a lot of ways to look at beige fat. You can expose mice to cold for several days, and they'll develop beige fat in their subcutaneous fat. Or you can give them daily injections of a beta-3 agonist for a week, and that will also have them induce them to form this beige fat. We use a beta-3 agonist, a beta-3 adrenergic agonist in these experiments because the beta-3 adrenergic receptor is exclusively expressed in mouse adipocytes, okay? And it's not expressed much in the rest of the body. So we did this experiment. You can see this is just staining for UCP1. You can see this is the vehicle injected mice. These are the mice injected with the beta-3 agonist. You see a big increase in UCP1 levels. This is a higher magnification. You see that these cells are multilocular, express a lot of UCP1. Well, what's happening to glycogen in this paradigm? Well, to our surprise, we found that glycogen, not detectable basically in vehicle-controlled mice in the subcutaneous fat, but a pretty big increase in glycogen in the mice that have been injected with the beta-3 agonist. And interestingly, the glycogen seems to come in the regions of the subcutaneous fat where you see a UCP1. Really in the same cells that are undergoing beiging, you see accumulation of glycogen. And we even did some EMs. This is one of the EM studies we did. Again, here's the control and the CL or the beta-3 injected mice. You see these particles of glycogen by EM. they seem to be coming around mitochondria. I don't know, nobody thinks mitochondria are important, but nevertheless, these things seem to be popping up around the mitochondria. I don't know why that is. It's possibly interesting. This is just, oops, I must have missed. Anyway, I have another slide, I must have missed it, but here's the quantitation of that data. Again, this is just our paradigm, 7-day injection of CL, big increase of UCP1. This is a message. Dia2 is another gene that's involved in thermogenesis. And here's the glycogen, here's the quantitation of the glycogen effect. Now, where's the glycogen coming from? Well, we just said, well, 7 days, maybe there's an induction of some of the genes involved in glycogen metabolism. And here we really saw a big surprise. There's like a cassette of glycogen metabolism genes that are coming up in response to the 7-day injection of a beta-3 agonist. GYS1 is glycogen synthase 1. This is a muscle isoform, it goes up. The liver isoform of synthase really goes up. This is really a surprise because most people, most of us, including in my lab, didn't think this isoform was even expressed in fat. PTG goes up, it's the name of PTG. And then the enzymes that break down the glycogen, phosphorylase, both the muscle form and the liver form. The muscle form doesn't change, but the liver form comes up a lot. This is at the mRNA levels. At the protein levels, it's even more impressive. Synthase, the muscle form, the liver form, phosphorylase, all come up in concert with UCP1 at the protein level and DIO2. So there's something really interesting going on here in these same cells. Glycogen, this cassette of glycogen enzymes is going up in concert with the browning of the white fat, of the subcutaneous white fat. So is this important? Well, we took advantage of the knockout mice that we had, and we gave these mice either vehicle or 7-day injection of a beta-3 agonist. And as you can see here, this is just looking at UCP1. This is looking at the glycogen levels. You can see, as I showed you before, the glycogen is elevated in response to beta adrenergic activation, and no increase in the knockout mice. Not surprising. This is what we saw before in liver. We know this is important. But if we looked at browning of the white fat, what we see is here's the wild type mice, big increase in UCP1 staining in the wild type mice. In the PTG knockout mice, browning was really deficient. So this makes us think that PTG or glycogen is really required for the browning of white fat. Quite a surprise. This is just a quantitation of the glycogen levels. You see the glycogen increase is wiped out in the knockout mice. And here's something interesting. UCP1 protein is induced, much less so in the PTG knockout mice, but the other genes in glycogen metabolism are okay in the knockout mice. We can still make synthase and the other genes that are involved in response to beta-3 activation. This is again the 7-day activation of beta-3. So this suggests that there's something selective about the transcriptional program in response to a beta-3 agonist. And we did RNA-seq on these mice. Again, so in this experiment, we looked at wild type and knockout mice plus and minus the beta-3 agonist, and then did RNA-seq. And the take-home message here is that there were lots of genes, 550 genes or so, that were induced by beta-3 activation in subcutaneous fat in these mice. Only 111 of them were different in the knockout mice. So there was a very selective effect on gene expression in response to beta adrenergic activation. And as we looked at the pathways that were involved, and I don't like doing this, but I'll just give you a kind of an overview. The thermogenic pathways were compromised in the PTG knockout mice, but other pathways that are activated by beta-3 activation, PPAR pathways, mitochondrial biogenesis pathways, and the glycogen pathways, with one exception, and I don't want time to talk about that today, were unchanged between the knockout mice. So there's a very selective effect of glycogen on this mice. Now, I know you're thinking, okay, Alan, you told us these were global knockouts. You just told us about the phenotype of PTG knockout in liver. How do you know this is really cell autonomous? So we made a conditional PTG mice and crossed these with the DIPO-Q mice to make a fat cell specific knockout. And we see the same thing, a reduction in the induction of UCP1 in response to 7-day injection of CL with no effect on the glycogen genes. And what about the biology of this? Well, we looked at, okay, that was all stuff done with 7-day injection. We also decided to do some cold exposure experiments. So here we did a 7-day exposure to cold. And you can see cold also gives a big induction. It's better actually than you see with injection of a beta-3 agonist, a big induction of glycogen. This induction of glycogen was blocked in the PTG knockout mice. Then we made brown and beige fat specific knockout of PTG by crossing our conditional mice with UCP1 cream mice. So now we're just knocking it out in beige and brown fat cells. And you can see UCP1 induction is still repressed in these mice. And we looked at cold induction of UCP1 in these mice too. It's repressed. So this is obviously a cell autonomous effect. And then we did an interesting experiment. We put these mice in metabolic cages and exposed them to cold over time. And what we see is that, so we equilibrated the wild type and the knockout mice in metabolic cages. We started to put them in the cold. You see they slightly start increasing respiration. Then there's this big boost about the 4th or 5th day. This is really when the beige fat is taking over in terms of respiration to combat the effect of cold. And what we see is that there's a big difference then in respiration between the wild type and the knockout mice. So glycogen accumulation is required for this cold adaptation over prolonged exposure to cold. And then we did one other experiment. We also wanted to look at a weight loss paradigm. Now, we know that we knew that knocking out PTG and fat didn't really have any effect on weight if we put these mice on a high-fat diet. As you can see here, the weights were identical. But if we induce weight loss in these mice by 7-day injection of the beta-3 agonist, you see we saw weight loss in the wild type and not in the knockout mice. So all the biology associated with beiging of fat is repressed. It's basically repressed in the PTG knockout mice. You need glycogen, and very surprising, right? You need glycogen to see the browning of white fat and all the physiological outcomes of that. And I'll just point out one other thing. We collaborated with 2 groups, with Jake Lucis here and with Michael Ryden in Sweden, to see if we could see any kind of correlation in humans. Now, we haven't done any experiments in humans, but we got hold of RNA-seq data from adipose tissue from patients. In Jake's cohort was a big collection of men. In Michael Ryden's cohort, a collection of women. And we can see there was a pretty strong correlation. Actually, for clinical data, it's pretty good. A correlation of some of these enzymes, glycogen synthase, the liver form, PYGM, with both BMI. It's a negative correlation with BMI and HOMA-IR. So it looks like metabolic health. I don't want to make too much of this, it's just an association, but metabolic health in patients is associated with the ability to express the genes associated with glycogen metabolism in fat. So to summarize, glycogen accumulates in IWAT adipocytes upon adrenergic activation, coincident with UCP1. It's near mitochondria. There's a cassette of glycogen metabolism genes accordingly upregulated. This is all downstream of PKA. If we block PKA, we block this induction. We don't know the precise pathway, but we know it's a PKA pathway. And we know that if we knock out PTG, we block glycogen accumulation and UCP1 induction and IWAT. And by the way, I'd be happy to answer questions about this. No effect in BAT at all. Brown fat, which has glycogen, is not regulated by this pathway at all. Just the beige fat. I'm not really sure why. And the other thing about it, it's very specific. There's no difference in catecholamine sensitivity in these mice. So lipolysis, for example, in response to beta adrenergic activation is completely normal. Okay, but it does prevent the weight loss, and there's a correlation of expression with metabolic health. So what's the mechanism? What is glycogen doing in these mice? Now I'm just going to spend a couple of minutes discussing what we think is going on. So what do we know about induction of UCP1 in fat cells? We know that the cyclic AMP pathway is involved, the PKA pathway is involved. Sheila Collins has worked on this quite a bit. She also has shown that P38, another protein kinase, is also required for this activation, probably downstream of PKA. Okay, it's not in parallel, but downstream of the the PKA pathway. We don't know the precise way yet, but that's happening. We confirmed these data. So we took adipocytes that were differentiated from mouse pre-adipocytes. We call these PPDIVs. They were differentiated in vitro. And these mice respond well to beta-3 activation. We gave these mice a beta-3 agonist. There's a big increase in UCP1 induction. And you could block this with inhibitors of P38 or PKA. This is already well known. We just confirmed this. But this made us wonder, because the PKA pathway wasn't influenced by knockout of PTG because lipolysis was okay. This made us wonder whether the other pathway was possibly involved, the 38 pathway. So how could this be? We took a look at activation of P38 in these in vitro cells that we made from the mice. So we made these cells, these adipocytes from both wild type and knockout mice. Then we gave these cells a beta-3 agonist and measured the activation of P38. As you can see, it was repressed in the knockout mice. So it looks like the glycogen pathway is somehow required for the full expression of the activation of this kinase. So the question is, what's the pathway? What's going on with the activation of P38? Is it glycogen? Is it AMPK like we saw in liver? It's not. AMPK has no role at all in this, at all. We knocked out AMPK in fat cells. It didn't have any effect on this pathway. Now, you remember, and you're probably thinking when I first told you about this cassette of enzymes that was induced by CL. First of all, you're probably thinking it was weird because at the beginning of the talk, I told you that epinephrine causes hydrolysis of glycogen. And now you're telling me that in the 7-day paradigm, it gives you this increase in glycogen. But I think I showed you why, because you get the induction of all these genes. But why would you not only induce the genes involved in glycogen synthesis, but also the genes involved in glycogen degradation, like GP and AGL, which I haven't talked about today? Furthermore, we know that glycogen, the enzymes that break down glycogen are activated by PKA, okay? So this made us wonder, wait a minute, maybe it's not the glycogen per se that's necessary for the activation of UCP1 induction, but it's the turnover of glycogen, okay? So maybe it's not glycogen, maybe it's a dynamic control of the turnover of glycogen. Since we know that hydrolysis of glycogen is getting activated by epinephrine in this system. So we wanted to attack this problem. And first thing we did was to measure the turnover of glycogen. The product of breakdown of glycogen is glucose 1-phosphate. We could measure it in response to an acute treatment. These are again in the in vitro system, in the adipocytes that we made from the wild type and knockout mice. You see there's this big production of G1P. This is the hydrolysis of glycogen in response to a beta-3 agonist in these cells in vitro. This is reduced in the PTG knockout mice, probably because the glycogen levels are lower in these mice. But then we actually gave these mice, or gave these cells, a phosphorylase inhibitor to block the degradation of glycogen. And we wanted to see what that did. And what it does is it blocks the induction of UCP1 in response to a beta-3 agonist. This is all done now in vitro, okay, in these fat cells. So you can see here, this is induction of UCP1, the time course in response to beta-3 agonist. You see it's repressed if we administer to these cells an inhibitor of the breakdown of glycogen. Okay, so the breakdown, it's the metabolism, it's the breakdown of glycogen that's required for the activation of UCP1, of the induction of UCP1 through the activation of the kinase domain. Now, what is the mediator of this? And we looked at glucose 1-phosphate. We thought maybe it's some kind of allosteric regulator. I won't bore you with all the negative data. The answer is ROS. Okay, when glucose 1-phosphate is produced, it's metabolized, it generates ROS. You can see the generation of ROS in response to a beta-3 agonist in these fat cells. And if you look in the fat cells derived from the knockout mice, the generation of ROS is reduced. If you add a GPI, an inhibitor of glycogen hydrolysis, it's reduced even more. Now, it's not completely reduced. And I'll tell you, if some of you are probably interested in this, There's a species of ROS that's cytoplasmic. That's the species that's reduced. There's a mitochondria. Who cares about mitochondria? But there's a mitochondrial species of ROS that's unaffected by GPI or by the glycogen level. So there's two things going on, but it's this cytoplasmic ROS that's involved in the induction of UCP1. And if we add a quencher of ROS to these fat cells, we can block the activation of P38, as you can see here. This is adding N-acetylcysteine, which is an inhibitor, a quencher of ROS. And this also blocks the induction of UCP1 in these cells and DIO2 to some extent. So this is kind of our model of what we're talking about today. What we think is happening is that in beta adrenergic activation, like you see in cold exposure, any other kind of catabolic stress of these fat cells, you get activation of these pathways. pKa, through mechanisms that we don't understand yet, induces this cassette of glycogen metabolizing enzymes, PTG, GS, GP, HEL, these guys all come up. This is something that we do not see in liver. So there's something unique about adipose tissue and adipocytes that respond in this way with these enzymes. We don't understand the differences. We know that beta adrenergic exposure or cold exposure also increases the uptake of glucose in these cells, mainly through GLUT1, which is an expression of which is increased. So now the glycogen starts to really accumulate in these cells. And now these cells are primed for a thermogenic response because pKa now can activate GP. This gives rise to hydrolysis of glycogen, the generation of glucose 1-phosphate, which is metabolized. And we don't really understand this yet. We're working on this to produce ROS, which is necessary for activation of this pathway that gives rise to UCP1. Now, what's the target of ROS in this pathway? P38 does not look like it's the direct target of ROS. So we think there's something else in the pathway. We're investigating this now. We have a few ideas, and I'm glad to talk about it later if anybody's interested. But we think this is the thermogenic response. So what does this mean teleologically? We think that glycogen is a fail-safe mechanism to tell the cell that it's okay to uncouple ATP. Now, uncoupling ATP is a serious business, right? You don't want to uncouple ATP unless you can really afford to. This is a way for the cell to say, we have enough energy on board. Let's uncouple some ATP. Let's make some heat. Okay, it's time to make some heat, and we can afford to do it. So glycogen is a cue to these cells to tell them it's a permissive cue to say thermogenesis is a good idea now. Okay, so this is my last slide. I just want to remind you what I talked about. I'm trying to convince you that glycogen is not boring. That glycogen is interesting. It's a signaling molecule. In the liver, it's involved in a forward loop that tells the cell to repress AMPK and sustain the activation of this mTOR-SRBP pathway. In other words, store energy as lipids. It's a signaling molecule. Store energy as lipids. And in fat tissue, it's involved in another loop that's telling the cell, we have enough energy, go ahead, let's make some ROS, let's activate UCP1, let's burn some heat, okay? So it sustains the thermogenic response. Opposite effects in the two different cells. And it suggests that glycogen could either be a good player or a bad player. And I think there's other information on this. And I think other things will emerge in other cell types too. There's a paper in Cell this week about glycogen in tumors. Actually, that was quite interesting. So let me just tell you about who did all this work. The work on fat tissue was done by a very talented postdoc, Omer Keenan, and he also was assisted by Joe Valentine, another postdoc in the lab. All the work in liver was done by Bin Liu, who was a postdoc in my lab at Michigan, and Bishen Zhang is also working now on the liver part in my lab in San Diego. And these are some of the people I talked about today that were involved in this work. We collaborated with Ron Evans, Michael Ryden, with Jake, Sushil Mahata, who's a EM guy, and Ed Chuchani at Harvard, who helped us measure the oxidation of proteins in response to ROS. And I'll just stop there and take questions. Thank you very much.
Thank you. This was wonderful. We have lots of questions. So we have also questions that are coming on the chat. And we'll
Should we
take
answer
after.
those too?
Maybe.
I mean, they weren't
We'll start
interested
here.
enough to come. I don't
So
know.
we'll start here first with students and postdoc questions. Yeah.
Adaptive role of glycogen, increasing glycogen in the high fat diet, where the was just happening as a.
Okay, can I hear that one more time? I'm sorry.
Do you think there's an adaptive role of increasing glycogen in liver in high fat diet?
In the fasting state, yeah, I think there is, and I didn't show you the glucose homeostasis data either. So there's a little more to it than I showed you, but I think it is an adaptive role. I think the question is there a pathological role in NASH for glycogen? So, you could think of glycogen as a buffer for energy, and it might be protective in some ways. And then if it gets too high, it might be harmful. So this is something we're investigating right now. We're trying to manipulate the liver to see if we can change the glycogen metabolism and see what happens to the function of the liver and to fibrosis and damage and things like that. So it's a really interesting area.
More questions, students, postdocs?
So, Alan, thank you for making glycogen interesting. It's kind of a running joke in my lab that I roll my eyes when people mention glycogen because I don't know what to do with it. So now we have some ideas. One thing you didn't mention is its potential use as a substrate for energy. So what happens if you impair fatty acid utilization in these cells, so then they switch to the glycogen that's around the mitochondria?
Yeah, we haven't tried to do that, and that's a very interesting question. So, yeah, the glucose production, yeah, right.
Which is actually an interesting question. What is actually the turnover rate?
Yeah.
And is that related to the fact that they are near the mitochondria?
We can calculate that. I don't know the answer to that. We could calculate that. We have been thinking about this a lot. In fact, we actually have been thinking, is there a feudal cycle of glycogen metabolism itself as a thermogenic process, like a non-genomic effect of glycogen on energy metabolism or on heat production or something like that. So, and maybe that's has something to do with why it's near mitochondria. Maybe the glucose 1-phosphate's going to mitochondria, possibly. So that's another thing we've been thinking about, but we haven't investigated this yet. So this is really just, we're starting this. By the way, I should have mentioned, by the way, that the studies on glycogen and fat were published, I think, I haven't looked at it yet because I haven't had time, but we think we're published today in Nature. It should just have come out.
See, you're just talking, it's published on the same day. That's amazing. Now, the reason I'm thinking about it this way is that if you're saying the two things are close to each other, those glycogen and the mitochondria, and I saw this mitochondria and I could tell that those mitochondria are moving mitochondria. They're not parked. So if they're moving together, which is very unlikely that they your little glycogen thing is moving with the mitochondria, or the turnover rate is fast enough that you're just seeing what these mitochondria were involved in making happen just there.
Yeah.
And that's why I was thinking about the turnover. But there is a question here.
I have two questions. The first question, you say that there is no accumulation on glycogen on the fat. It means the turnover is more efficient.
In brown fat.
We can repeat the question.
Okay. Yeah. So yeah, it's a very good question. So the question was, I said something that there's no turnover and there's no regulation either of these enzymes in brown fat. And why is that? And so I would say that we didn't really see any control or any regulation of. There's a lot of glycogen in the basal state in brown fat, and it may be that there's enough there to respond. I mean, the glycogenolysis is probably happening in brown fat in response to beta activation, but there's not a priming effect like we see in beige fat and brown fat. So one thing you could just say that there's just enough glycogen there in the basal state to make all the responses that you need. We we don't see a big transcriptional upregulation of UCP1 in brown fat in response to cold because there's already a lot of UCP1. It's not like beige fat where you see a big increase. There's a little, kind of a little increase. So I think the regulation, the transcriptional pathways that are regulated in brown fat are not quite the same. I think in brown fat, the regulation is more dynamic and metabolic than transcriptional in response to cold too.
What happened in UCP1 knockout mice?
It's a good question. We haven't,
Repeat the question.
the second question, what happens in UCP1 knockout mice? And that's a very good question. And we haven't done that experiment yet. And that'll actually answer this question is there a non-genomic effect of metabolism of glycogen with respect to heat? So it's a very good question.
It's actually bringing in her mind. There's this question has another layer, which is if it activates energy, it relates to energy expenditure, like you say. Okay, now I have glycogen. Let's spend energy. Not all cells have UCP1. Maybe what about kidney? Kidney has glycogen. Can kidney do that? So, and then there are other uncoupling mechanisms. One of them was discovered by her. And so could it activate uncoupling in other cells? You could see that by respiration, probably in using seahorse that you have.
Yeah, I don't know. It's a good question. Very good question.
Mark.
Yeah, I found most interesting the connection in the liver with glycogen. So I was thinking, could it be the effect on the liver side? Could you get it by look for more glycogenesis, making more lipid droplets, which is also energy consuming? And also we know that differentiating white leukocytes, they have the ability to oxidize fatty acids, right? So some of the component was coming by the need of making these new lipids to have more lipid droplets.
Yeah. Yeah, it's a very good. So I think that I think the question from Mark was is there an interplay between glycogen metabolism and lipid droplets and the regulation of the lipid droplet through all the metabolic events that go on. And I have to say that we didn't see any gross changes, but we didn't really look carefully at this question. And we didn't certainly do any dynamic experiments to study the metabolic flux of anything with respect to this. So yeah, it could very well be that there's more going on. Also, it could be a difference between short and long-term adaptation mechanisms that go on. We haven't looked at that. Yeah, through the glucose. Yeah.
Okay.
Since I'm the macrophage biologist, I've asked this question. So we know that in addition to the pathways, I think lysozyme calcium for many years was shown that macrophages can also make glycogen. So are you interested in looking at it?
Yeah, that's a very good idea. And actually,
What's the idea?
I mean, he asked me about the glycogen pathway in macrophages. And of course, that's a very good idea. We worked a lot on this. And we did have a paper about glycogen accumulation controlling inflammation in general and in adipose tissue a couple of years ago. That was mostly on the glycogen and fat, though, not on the glycogen and macrophages. So yeah, glycolysis obviously is really important in macrophages, and this could be an indirect way to regulate that. Actually, I don't know which of the targeting proteins are expressed in macrophages, or if there's a difference in subtypes of macrophages. But you might expect to see that, right? Because they are either glycolytic or not. So we haven't looked at this at all. I haven't thought about this actually, but it should be very easy to do the knockout of PTG if it's expressed in macrophages in our, from our mice. So it's a really interesting idea. We haven't studied the metabolism of macrophages as much. We don't study it like we used to a few years ago, but it's a really good question.
Anthony.
Yeah, we haven't done that. So Peter Roach has done a synthase knockout and looked mostly in liver and seen the expected effects. We haven't done that at all. So we've just focused on the targeting protein.
Anton?
In your electron microscopy images of the adipose tissue, you can see glycogen grams per mitochondria. Is there a difference between mitochondria that are attached to the droplet versus those?
Yeah, I don't know. We haven't looked at that. So, I mean, I'm happy, any of you guys want to see those EMs, I'm happy to show them to you.
That would be really interesting. So Rebecca, that is sitting just behind him, was together with Anton, we're recently showing that the main change that you see in mitochondria during browning is happening on those mitochondria around the lipid droplet. So now that you're talking about browning and how important the glycogen is, so the question is, are those specific type of mitochondria or the others, and they have different fuel preference? That could be interesting just to look at these EMs again.
Yeah, and these are multilocular cells now because lipolysis is getting activated. So yeah, there could be all kinds of interesting differences. Yeah, we haven't thought about that.
So somebody else raising hand. I think we have also a question here. If you click on the Q&A and probably in the chat as well. Is there a question there?
Could PTG manipulated genetically for obesity? Yeah, or for NASH, actually. It's a good question. I guess, I don't know. I guess so. We haven't thought about that. You know, one idea that we have thought about is that PTG is such a versatile molecule because it binds to 3 different, actually more. I didn't talk about AGL very much today or other things, but it might be possible to manipulate the interaction of PTG selectively with some of its partners. And that might be a way to get things to go in one direction as opposed to another, or in one tissue. So yeah, it's interesting.
Any more questions? Wait, in the chat, is there a question? No.
Glycogen metabolism is known to go down in diabetic brain. Is there a connection with brain fatty acid oxidation and connection with UCP1 activation in diabetic brain? Yeah, it's a good question. I don't know. We haven't looked at this, and I'm really trying to find somebody to look in brain, because I have this feeling that glycogen might be toxic in brain, and maybe even involved in long-term pathology in brain. So it's a really good question. We haven't looked at all. PTG is definitely expressed in brain. It's really expressed in astrocytes, and its expression goes up in response to cyclic AMP in those cells, just like it does in fat cells.
You have a new question who came up on the Q&A behind this one.
I think that was the one. Oh, there's another one now. Okay. What is the percentage of glycogen synthesis that's controlled by hormones versus substrate flux? Well, substrate fluxes are also controlled by hormones, so it's really hard to answer that question. But Kai Sakamoto did this experiment where he knocked an alanine into the GSK3 site in synthase to really kind of figure out how important GSK3 inhibition was with respect to activation of synthase. And it turned out to be not that important. So he thinks that either PP1 activation or allosteric control of synthase is more important. But I don't know. It's hard to interpret those data still. I'm not really sure, because it could be that if you overwhelm the system with an allosteric substrate, it could be dominant, but it doesn't mean it's always more important. All right, so.
Okay. With that, we want to thank again. Great talk and being the pioneer of being an in vivo speaker. And I want to also thank Dion Baybridge, who has really helped us set it up and make it happen that we are coming back together here. Dion Baybridge is working with the dean, and he has made it possible for us to do that. Thank you.